Hello, welcome to a Zoominario. We're very excited today to have Geo Santiago-Martinez. Geo is a Mexican microbiologist interested in astrobiology and microbial ecophysiology. He got his PhD from UNAM, Universidad Nacional Autónoma de México, and then he was a postdoc in the Ferry Lab at Penn State University. Geo is an assistant professor at the University of Connecticut. His research focuses on mechanisms of energy conservation and cellular processes in oxygen-sensitive microorganisms and their function in microbiomes associated with extreme and non-extreme environments. Thank you so much, Geo.
Yeah, thank you so much for the invitation, and I'm very happy to be here. So I started my lab last fall, so I'm a new PI. So I'm the PI of this lab, the microbial ecophysiology lab. So we work with different microorganisms, but today I will talk about methane-producing microorganisms. So if you have any questions about my talk, my research interests, my future plans, or anything that you want to ask or know, you can send me an email, or you can follow me on my social media, X for my call like Twitter. But yeah, so feel free to ask any question at the end of this talk, or send me an email at any time. Okay, so the microbial ecophysiology lab at UConn, we work with different microorganisms, but we now are focused on a small group called methanogenic archaea, or methanogens, or methane-producing microorganisms. So these microorganisms are part of the domain archaea, and they have some different characteristics in comparison with the members of the domain bacteria or eukarya. So, and that is the main classification of microorganisms. So in this archaea domain, we can find the methanogenic archaea, or methanogens, or methane-producing microorganisms, and they have three main characteristics. The first characteristic is these microorganisms produce methane using a methanogenesis pathway to produce energy and produce also the biomolecules necessary for the cell duplication. The second characteristic is these methanogens are very sensitive to the oxygen and very sensitive, and the third is they are very sensitive to the light. So we have to grow them in anaerobic condition and dark condition. So that is the reason why these microorganisms live in very, very specific environments, but in very diverse environments. So we can find, for example, methanogens in the coastal, in the sediments of coastal sediments, or we can find, for example, in sediments of aquatic environment, freshwater environments, or marine environments. But also we can find methanogens living inside of different animals or plants. So that is the reason why we are very interested in understand the metabolism of these microorganisms. So, and these microorganisms are very essential for the regulation of, of the global carbon cycle, because they perform the degradation of the organic matter in anaerobic condition, in oxygen-free environments. So, for example, the degradation of this biomass for bacteria, fungi, or other microorganisms produce these very, very small molecules like carbon dioxide and hydrogen, or acetate, or other compounds with methyl groups. And they can, and those are the perfect substrate for methanogenic archaea to produce energy and to produce methane. So the biological production of this methane release a lot of methane to the atmosphere, and it has an implication in the ecology of our planet, because this methane is a greenhouse gas. So because the methane is a greenhouse gas, it has a huge implication or role in the climate of our planet. But also we can use this methane as alternative biofuel for anthropogenic activities. So studying this methanogenic archaea, we can have these different applications, like ecological applications or biotechnological applications. So these microorganisms are very important to continue with the global carbon cycle, and we can, my lab is focused on understanding the metabolic pathways and the ways that these microorganisms can survive in optimal conditions, but also in extreme conditions. So in the microbial ecophysiology lab, we work at three different levels. So the first one is proteins. We can work with biomolecules involved in the anabolic pathways or energy conservation mechanisms. The second is we can cultivate the cells and we can evaluate some physiological parameters. For example, evaluate the metabolism of these microorganisms, the preference for energy sources and carbon sources, or any other type of conditions. For example, cultivate these microorganisms in oxygenic conditions because they are sensitive to oxygen. So we can evaluate different stressors. And the third level is we can work with communities, with microbial communities. So we have some examples. We have some projects in the lab focused on the microbiome analysis of different environments. And the idea is to understand the role of this methanogenic archaea in different environments, but also the interactions between microorganisms, but also hosts and microorganisms. So we have some species, we can cultivate some species in the lab. So we have these anaerobic chambers, a special infrastructure and equipment to make the media and grow the cells in oxygen-sensitive, oxygen-free environments. So we usually make the media inside of the anaerobic chamber. So in that way, we can eliminate the oxygen from the air and we replace that gas with nitrogen and carbon dioxide. So we use these bottles to grow the cells. For example, here you can see this is the media. This is a culture. So we have a liquid media, and we have here some bubbles, which is an indication that the cells are producing methane. So it's very funny to work with these installations because sometimes I will say that this is funny, but sometimes it's very challenging because it's very difficult to eliminate the oxygen from the environment. So that is the reason why we have to use a specific or very specific conditions, so very specific equipment to work with these microorganisms. So I have been working with different species, but my favorite microorganism is Methanorthocyanine acetivorans. So this is a microorganism. This microorganism was isolated from the coastal sediments of close to California. So in that environment, the degradation of this microalgae produce acetate, and that acetate is the main energy source for Methanorthocyanine acetivorans to produce energy and to produce methane. So we can cultivate these microorganisms under laboratory conditions, and we can evaluate different conditions, or test different hypotheses, for example, some stresses. So, and it has been proposed as a model organism to study, for example, methanogenesis pathways or other anabolic pathways. So we can use different approaches. For example, we use comparative genomics to evaluate or to create hypotheses about the potential function of some genes. For example, one project that we have in the lab is we know, for example, these Methanorthocyanine acetivorans can use the acetate from the environment to produce energy and produce methane. So, but we don't know how this microorganism use that acetate. So we have other examples. For example, in mammalian cells, we can find this system in which this protein is a transporter. And here you can see, for example, this transporter can incorporate or help to incorporate this monocarboxylate, in this case lactate, but it could be used other monocarboxylate like acetate. But in this mechanism, this other protein, carbonic anhydrase, use or produce this proton, which is essential for the incorporation of this monocarboxylate. So in this mechanism, we have to have this transporter, this other protein, producing this proton for the incorporation of this monocarboxylate. So we have similar genes encoding proteins potentially involved in the incorporation of acetate in Methanorthocyanine acetivorans. So here, for example, we have in Methanorthocyanine acetivorans, we have genes for the genes potentially encoding an acetate transporter, which is similar to this. And we have this carbonic anhydrase, which is a protein similar to this. So we have been studying the molecular mechanism involved in the incorporation of this acetate. So we can make different experiments, for example, growing the cells, making mutants with the no ability to produce these proteins. So we are trying to understand the incorporation of acetate, but also the mechanism in which acetate can be degraded or can be used for this mechanism to produce the energy and produce the acetate. But other very fantastic characteristic of this mechanism is that this mechanism, methanization acetigrans, can produce a biofield against different types of stresses. For example, here you can see, because these mechanisms are very sensitive to the oxygen, when they sense oxygen, they can produce this biofield. And this biofield is like a barrier for protecting the cells against this stress. But we know that they can produce this biofield, but we don't know the molecular mechanism, and we don't know actually the chemical composition of these biofields. And it's very curious because these mechanisms also can produce a biofield against stress induced by heavy metals, for example, cadmium. So here, for example, you can see a culture, and this is the biofield, and these bubbles are indicating that these cells are alive, living inside of this biofield, and they are alive because they are producing methane and carbon dioxide. So this biofield is a way to protect the cells from stress conditions or from the stressors. But okay, but I have this expertise in the physiology, in the microbiology of these microorganisms, but from the marine environments. So right now, my lab is also interested in working with other type of microorganisms, because in the last years, many other people have found methanogenic archaea and other archaea inside of different animals and some plants. So if you can, for example, you can see here that some type of archaea living inside of these animals. And the most common microorganism is Methanobrevibacter smithii, which is a methanogenic archaeon. And now we're interested in understand what is the role of this microorganism inside of different hosts. So, for example, these animals, herbivores, they are very, very famous because they produce a lot of methane, and they produce methane because they have a lot of methanogenic archaea as part of their gut. So they produce a lot of methane. For example, a cow can produce this amount, 500 liters of methane per day, and humans produce just 0.4 liters of methane per day, which is very small amount, but we are producing methane. And we are producing methane because we have methanogenic archaea as part of our microbiome. So, but we don't know the function, or we don't know the energy sources for this methanogenic archaea inside of our body. So that is the reason why my lab is right now working with using this microorganism, Methanobrevibacter smithii, as model organism to study, for example, the preference for energy sources and other mechanisms, or other or grow these cells under optimal conditions, but also in a stress condition. So in general, in the field, we have the idea that the interaction between bacteria and archaea is important to maintain the health of humans and other animals, because bacteria produce carbon dioxide and hydrogen, and those are substrates for methanogenic archaea to produce energy and produce methane. So this balance, this interaction between bacteria and archaea is important to maintain the health. So, but actually that is a hypothesis. So now we want to know the molecular mechanisms or the metabolic pathways in this microorganism, and then we can use different other models, for example, using synthetic communities to evaluate the interaction between bacteria and eukarya, bacteria and archaea, or other microorganisms, to try to understand these relationships inside of the microbiome, the human microbiome especially. So the only problem is that we are this microorganism is produce very, very low biomass. So right now we are trying to test different grow conditions because we think that maybe this microorganism, because we have the genetic information, maybe this microorganism using other energy sources. So maybe this microorganism is using not only using carbon dioxide and hydrogen, but maybe it's using also acetate, formate ethanol, methanol, or other molecules to produce energy and produce the biomolecules necessary for cell duplication. And this is because in the human gut, the concentration of these other compounds is sometimes higher than the carbon dioxide and hydrogen. So we are testing these different conditions or different preference for energy sources in our lab. But one other basic question that we have in the lab is that how we're getting archaea. Because, for example, we know humans or other animals are producing methane because we have a methanogenic archaea inside of us or a body. So, but we don't know how we are getting the archaea, especially methanogenic archaea. So I have this collaboration with Dr. Sharifa Crandall and from Penn State University and Timothy Miles from Michigan State University, because they have this expertise in different fruits, for example, grapes. So they have this expertise in analyzing the microbiome composition of these grapes. And we started this collaboration because I want to know if these grapes or other food, I want to know if we can get archaea from the food. So when we started this collaboration, we found that the microbiome of the grapes is composed by fungi, bacteria, but also archaea. So we found two different types of archaea. One is this, which is a methanobacterium, which is a methane-producing archaea. But also we found which is essential or important in the carbon cycle. But also we found this other archaea, ammonia oxidizing archaea. So, and this is important in the nitrogen cycle. So in general, right now, the explanation that we have is that these microorganisms are important for maintaining the health of these fruits, because we only found this archaea in healthy grapes. But actually, we don't know the role. So we have to do more experiments, and maybe we can try to find archaea in other different microbiomes from natural environments or maybe from food. So we have this expertise in the lab. We have this expertise in working with methane-producing archaea, but right now with this new area in expeditions to other planets outside our planet, so people is finding, for example, these cycles of production and consumption of methane. So we are proposing to use a methanogenic archaea as model organism for astrobiology projects. So I'm not saying that this archaea is living in Mars. It's just like we can simulate the conditions from other planets and do some experiments in laboratory condition to try to understand, let's say, the limits of the life, or how the life from our planet can or evaluate if the life forms in our planet can survive in this extreme environment. So we're working in different projects. So our lab, I will say that our lab can have, working with methanogenic archaea, we can have the biotechnological approach, biomedical approach, ecological, and astrobiology approach. So I'm almost done with my talk, but this is just some important message to give regarding to the methanogenic archaea. So methanogenic archaea are oxygen-sensitive microorganisms. They are a very specific group of microorganisms with this characteristic, but it's a very diverse group of microorganisms. So we can find methanogenic archaea in different environments, extreme environments, but also in non-extreme environments. And we can find microorganisms living in different environments, like aquatic environments, but also living inside of different hosts, like animals or humans. And the main characteristic of these microorganisms is they produce methane using methanogenesis. So only archaea can produce methane by using methanogenesis pathways. Because, for example, other bacteria, some bacteria can methane, but they don't use methanogenesis pathways. So only archaea can use methanogenesis so far. And studying this potential with this type of projects, we can found different applications. For example, producing molecules important for biomedical application or biotechnical application. And we can provide new information about how these microorganisms survive in optimal conditions, but also in stress conditions. So it's just to say thank you to my lab, because I'm just a PI. So they are my students, they are doing the experiments. So they have this huge motivation to understand this microorganism. So we are starting the lab, but we are getting some very good results. And some of them, they are looking for other options. I mean, to join grad school or something like that. So I'm very proud of them. And yeah, that is everything about my lab, my new lab, my research interest. But something that I want to share with you is like, I'm from Oaxaca, Oaxaca, Mexico. So this is just to make some promotion about this place, because of course that is my hometown. So we usually say that Oaxaca lo tiene todo. That means Oaxaca has everything, because I would say that this is a very good place to visit if you want to know about the culture of different different type of foods. So, for example, in this slide, I try to represent the most important part of Oaxaca. So Oaxaca is in the south of Mexico. So this is a place with high diversity in terms of the culture, the food, and Oaxaca is very famous, for example, for the chocolate and the cheese, which is an special type of cheese. So if you want to visit some place in Mexico, I highly recommend to visit Oaxaca. And if you are looking for different food, or if you are looking for different environments, for example, if you like hiking or visiting different places, Oaxaca is the perfect place to visit. So, and also we have this very special word, Galaguetza, which means offering or sharing something. So this is, I will say that this is a very original tradition from Oaxaca. So that is something that we usually do. So because I'm from Oaxaca, I usually give something to my friends or my family or other colleagues. So when we are doing that, we say that we are giving Gelaguetza. So that means we are sharing something very special of us. And but also Gelaguetza means a festival. So something very important, or a very important event in Oaxaca is this Gelaguetza festival, in which many people can join this festival, and we can share. I mean, the people living there, they can share this different perspective, different culture, different food. So I would say that Galaguetza is a very a key word from Oaxaca. So I wish you have the opportunity to visit Oaxaca in the future. So yeah, so that is everything that I want to talk with you today. So if you have any question about my talk, or if you have any question about Oaxaca or about other different things, feel free to ask now, or send me an email or contact me at any time. So thank you so much for attending this talk.